Alright, a quick little recap on our 5 ways we can classify and ID organisms. Again, I kind of previewed some of these in lecture, but I'm going to go over them just a little bit more detail. And so our number 1 way we can classify and ID a microorganism is, yes, based on its physical characteristics. We can use it to help ID microorganisms based on their shape, based on their gram stain. We can see their colors, we can see their shape, we can see what they look like growing on a plate, looking at difference in colors. We can see what their colonies look like, what color are the colonies. We can use all these types of physical characteristics to ID out organisms. We can also look at larger organisms like protozoa, fungi, algae, and even worms, just looking at some of their outside physical characteristics. I'm like, this is looking at their colonies and some distinct appearance of colonies. As some of you are seeing in lab this week, that colonies can look very different from one another. The second way we can help ID and classify organisms is through biochemical tests. Again, a lot of organisms, especially bacteria, can look a lot alike. So then we start running various types of biochemical tests to help sort them out and organize them. Now, the basics of a biochemical test is that we give them a substrate. We're going to give these bacteria or microorganisms something, and we want to see if the microorganism can break it down, if they can use it, and if so, what will be the product. Now, generally, the product will turn some type of color so we can see that a product was made. And so this is showing, and we're going to do several biochemical tests this semester later in the semester, but we'll do some. This is showing that, yes, there's an organism in here, but whatever the substrate was, it didn't do anything with it. It stayed just this kind of reddish brown color. Versus another bacteria, another microorganism, may take the substrate and turn it into a product, in this case an acid, and all of a sudden it changed color. There was some type of pH indicator that shows there's an acid present now. And then another organism can take that substrate, make an acid, and not only does it make the acid, it also produces gas, which they're trapping inside this little inverted tube. And like, there's lots and lots of biochemical tests that we can run. Again, the basics is, if we give the bacteria a substrate, can it do something with that substrate? And if it can, what does it do with it? And those positive, negative results help us narrow down what type of organism it is. Now, and I'm like, tubes can get kind of putsy and they can take up a lot of room, and so there are actual machines out there that can run a whole bunch of biochemical tests all at once. It's called a microscan. Yes, it's scanning for various biochemical tests. This is showing a plate, each one of these little wells, and I'm like, it's a big plastic kind of plate, and there's little indents in it called wells. I'm like, each one of these is going to test for some other type of biochemical reaction. And again, looking for color changes to show that something happened, or if it didn't, and based on the color, what actually happened. And then we actually have computers that can read the colors to tell us what are positive and negative reactions that are happening. This is an actual picture of one of these biochemical testing machines. It's called the Vitek. This one's at Gunderson. And again, it can read all of these different types of biochemical plates, just looking at color changes. A third way we can help ID and classify organisms are serological tests. Now, the reason why we call it serological, the sero comes from serum. Now, serum is blood plasma without the clotting factors. Now, really what we're looking for in a plasma type substance are the fact that there are antibodies in there. And so we're really looking to see what kind of reactions antibodies are going to have with antigens. Now, if we are trying to ID out an organism and we put in the appropriate antibody, meaning the antibody matches with the correct antigen or substance, it will bind. And because every antibody has 2 arms, they look like Y's, they have 2 arms, they can bind to 2 different antigens or 2 different organisms. And as you start adding lots of antibodies, you start to get these big clusters of antibodies attaching to various antigens, because one antigen can be attached to by multiple antibodies. And so we actually see clumping. And so I'm like, if it's not the correct antigen, because looks different than this one, it won't bind. And so by adding antibodies, we can look for clumping to say that, yes, this organism is present because it has the correct antigens, in this case, little tiny triangles, and I'm like, versus little tiny squares on the outside, and we can physically see a clumping. A very common place that you would see this type of test, it's called agglutination. The glue means clumping or gluing together. I'm like, if you've ever done blood typing, if you've taken advanced already, you've done it. If you haven't taken advanced yet, or you are in advanced right now, you will be doing blood typing. And you're looking for this clumping. It just is antibody attaching to antigens. It can help us ID out organisms. Now another type, or a fourth way we can classify an ID organism is called phage typing. Now, phage typing, I should make note, and I'm like, is named for what are called bacteriophages. Now, even though a bacteriophage has that prefix bacterio in it, a bacteriophage is a virus. So a bacteriophage is a virus that infects a specific bacteria. And so what we can do as we start to ID out different types of viruses is that we can put unknown bacteria on a plate. So each one of these little tiny wells is an unknown bacteria. And we can add known viruses. And so we can look to see which bacteria the viruses infect. Viruses are specific for what bacteria they will infect. And so we can look to see it. I'm like, if you can notice that, these particular little circles of bacteria growing, kind of like big colonies, are starting to break apart, that is showing you that those bacteria are dying because they have a virus that is infecting them. And so we can, by having these unknown bacteria but known viruses, we can be like, well, because these particular bacteria are starting to get broken down or infected by viruses, they are most likely this group of bacteria. And then the last way we can help ID and classify organisms is, yes, to look at nucleic acids. Now, generally when we're looking at nucleic acids, the top two nucleic acids we're looking at are guanine and cytosine. They've noticed that similar organisms have the similar number of cytosine and guanine. And again, yes, G pairs up with C's. And so I'm like, you should have the same number of guanine and cytosine in an organism, but they know that similar organisms have the similar quantity of guanine and cytosine. So similar organisms share similar characteristics. One organism has 72% of all of its genetic information is the G's and C's. And I'm like, well, another similar organism would also have about 72%. I'm a completely different random species, bacteria versus human, and I'm like, might have 42%, I'm like, a lot different. So they can look specifically at that nucleic acid sequence. Now, we're not going to do that. If you take some molecular bio classes, then you can start to play around with an ID out and separate out these different nucleic acids. But for our purposes this semester, we're not going to. And then again, the rest of the PowerPoint is just looking at those dichotomous keys or flowcharts. So that's it. That's all I wanted to get through for the rest of this PowerPoint.